and it's my honor to have this interview. Thanks for the introduction. That's very nice of you to invite me. First of all, I would like to self-introduce myself. My name is Pingping and from School of Dentistry at UQ, Australia. What Ariel said, my research focus on oral diagnosis and therapeutic, especially using epigenetics. Regarding my career journey, originally I'm from China, of course, and I came over to Australia in Queensland University of Technology, which is QUT, for my PhD degree after my master's in China. During my PhD was focusing on using wind canonical pathway or some other biomaterials to promoting periodontal regeneration, which is part of the tooth root structure. After my PhD, I studied my first postdoc research fellow in one of the UQ leading institute called AIBN. My first postdoc was focusing on using nano materials to modulate how the mesenchymal stem cells sensing the surface. And it's totally out of oral biology. However, after my first postdoc, I was awarded a UQ fellowship, which allowed me to explore the epigenetics in osteoporosis and oral disease. And from 2018, I moved from AIBN to UQ School of Dentistry. That's when I really started my oral biology, especially in extracellular vesicles, research in periodontitis. That's my basic career journey. Since 2018 until now, I have been acting as a junior group leader within the school and under the supervision of my supervisor, Professor Sasho Ivanovsky. Regarding the second question, how the research interest focus my oral biology. So as I mentioned, my PhD was in oral biology, which is periodontitis treatment. Now my second postdoc was about oral disease. Periodontitis. Basically, I've been working on periodontitis. And I learned extracellular vesicles from one of my collaborators in UQ as well. And he's one of the editors in this journal as well. Associate Professor Carlos Solomon. I learned my basic and knowledge of EVase from him. And I think periodontitis is a very interesting research area for me and my small group. For the general audience, not everyone knows about periodontitis, what periodontitis is, why we are doing periodontitis. I would like to start with that. Periodontitis is a severe form of the gum disease. Basically, it starts from when you brushing your teeth, you realize, oh, my teeth are bleeding. That's the very early sign of periodontitis. That's the early stage of periodontal disease. We call it gingivitis. Unfortunately, about 95% of adults have gingivitis. We always have bleeding gingiva or bleeding gum. If you leave it untreated, some of the patients will progress from gingivitis to periodontitis. Gingivitis is so common among the whole world, not only US population, but it's the whole world adults, more than 90%. However, it's reversible. If you brush your teeth and prevent the bacterial invasion, your gingivitis can be reversed to be healthy. You will have a healthy gum. But around 30% of gingivitis patients will progress to periodontitis. Like you mentioned, periodontitis will lead to whole periodontium tissue loss. It has different stages, early stage and late stage, severe stage. We call it stage 1, 2, 3, 4. For the stage 4 patients, they're all gum a regression, which means you wouldn't have too much gingiva on your tooth, and you will see a tooth root if you see it from your own mouth or something like that. That's the only clinical sort of characterization of periodontitis. In the histologically, like you said, periodontal soft tissue, hard tissue, for example, alveolar bone, which is jawbone, you're losing the jawbone at least 20 or 30% or 50%. And you lose the gingiva, which is gum, and also the cementum, which is on the surface of your tooth root. This disease is not reversible. Very unfortunate. It can't reverse it back to healthy. That's why once you have the periodontitis, you need to go to the periodontist to treat your disease. Current periodontal treatments, there are two types of periodontal treatment for periodontitis patients. First one, and the start stage of treatment, is called non-surgical periodontal treatment, which is also short for NSPT. Basically, it's basically the dentist will put a probe and some other tools under your gum, skew all of the plaque, bacteria, all the tissues, which cause a lot of macro inflammatory macro environment. By removing the plaque, your gum environment is supposed to go back to less inflammatory environment. This NSPT is the first stage. The second stage will be surgical treatment. However, you wouldn't want to reach surgical treatment stage. That's why a lot of research are using the effect of non-surgical treatment on, for example, on epigenetic changes during those patients or some other gene expression profile. As we mentioned so many of periodontitis, there's funny thing about periodontitis is it's initiated by the bacteria. This whole disease, gingivitis and periodontal disease, gum disease, is initiated by the bacteria. In oral environment, we have more than 700 or 900 different species of bacteria, no matter you're healthy or you're not healthy. This is very striking for most of people. However, it's the truth. As any other organs, in oral environments, you have good bacteria, you have bad bacteria. For example, in periodontitis, we call a certain bacteria as periodontitis or periodontal pathogen. The key one was P. gingivalis. P. gingivalis was one of the key red complex which initiate periodontitis. However, more and more research realized that the bacteria factor is only less than 10% to lead to the periodontitis. All of other factors, for example, environmental, epigenetics, however, the whole pathogenesis or exactly what caused that, we are still researching on it, of course. A little bit strict away from periodontal treatment. As I mentioned, my research is focusing on periodontitis treatment and diagnosis. For diagnosis, we are using extracellular vesicles in our oral fluid, for example, saliva, or there's another fluid called gingival crest, GCF. It's under your gum. There's a fluid like very less, like few microliters of GCF. For diagnosis, in the aspect of us diagnosis, the current clinical diagnosis was mainly using a probing, which is a manual probe. The periodontist diagnoses a patient, put the probe under the gum to see whether you have deep pockets, which means to see the distance between your tooth root and to your cementum. To your enamel, which is if you have a deep pocket larger than 6 millimeter or 4 millimeter or 5 millimeter, then you have periodontitis. If your periodontal depth is smaller than 3 millimeter, then you are healthy. This is how our specialists are determining a patient whether it's healthy or not. Regarding this current clinical diagnosis, it basically, those periodontal pocket means that the tissue destruction already happened, which determine the history of disease. However, you cannot tell what's your current level of your periodontal health. That's why we are trying to use molecular level of biomarkers, for example, mRNA or DNA or even epigenetics, but in saliva. Saliva diagnosis itself is a research area, very hot surface research area in all of the disease. I'm sure you already know that saliva can be used to diagnosis for oral disease, for example, periodontal disease or dental caries. And at the same time, it also can detect it for systematic disease. A lot of researchers are using saliva to diagnose, for example, heart disease, or cardiovascular disease, or even some brain disease and other disease, and diabetes, for example. But most of us just ignore how powerful saliva is. Especially saliva can contain our local environmental factors as well. Although compared to blood, saliva has very low concentrated molecules. For example, for protein, it's 500 times lower than blood. However, it has everything what we need to test. Or one of the focus will be we're using extracellular vesicles from saliva. I don't need to introduce too much about extracellular vesicles because our journal is about this. So extracellular vesicles can be found that circulating in saliva. Those EVase can be from host human cells, or it can be from bacteria, or even virus or fungus everywhere. That's why one of my key focus of the periodontal diagnosis aspect is to using saliva EVase to see whether saliva EVase can be the biomarker to diagnose periodontitis, gingivitis, in healthy patients. So far, our research has covered a few aspects. We have found some microRNAs from saliva EVase, at least 3 microRNAs totally distinguished, expressed in the periodontitis compared to healthy and gingivitis. At the same time, our research also showed a DNA methylation, global DNA methylation can be potential biomarker to diagnose periodontitis. One of my PhD's project is working on the bacteria-derived EVase in diagnosing periodontitis. I was talking about a little bit too much about diagnosis. However, we are doing periodontal treatment using EVase as well. One of my PhD projects is a PhD student's project was using our local cell-derived EVase to treat this severe periodontitis disease, either in vitro and in vivo at the same time. Yeah, the role of EVase in periodontitis, like I mentioned, is EVase circulating in oral fluids for us, my interest is saliva and GCF. Those specific signatures of EVA cargoes, as I mentioned, microRNA global DNA methylation, even EVA particle numbers itself can be potentially a diagnosis marker. However, my own research at the moment, only pilot study, we only have around 20 patients each group. To further confirm the role of EVase in periodontitis diagnosis, that I need a bigger sample size, up to 100 or 500 patients. However, from our current limited sample size, I can conclude that we have found 3 microRNAs, a global DNA methylation, and periodontal pathogen-derived ONVs can be potential biomarkers for periodontitis, especially for the bacteria-derived EVA, which is of great interest of all of the field. However, the bacteria-derived EVA, we mainly focus on gram-negative bacteria-derived EVA, which are called out-membrane vesicles. ONVs are relatively new in our field, especially for diagnosis. When I searched the in-literature review, everyone was using single species of bacteria and culture the bacteria and then collect the ONVs and then investigate the cargoes and some other functional analysis. However, directly isolating ONV from biofluid, for example, saliva, is very rarely limited in the field, which needs more research to be done in the periodontitis. In that case, from saliva ONV in periodontitis is the role, I'm not sure, I don't know, because it's relatively unknown. However, from the current literature, P. gingivalis derived ONVs and some other periodontal pathogen derived ONVs from single bacterial culture, they are proven that have most of the pathogen components from the bacteria themselves, from the parental bacteria. This can be a great indicator that bacteria-derived ONVs are great biomarkers for periodontitis. This is the first aspect of periodontitis diagnosis. For treatment, EVase, the role of EVase in periodontal treatment. Luckily, my students, we recently published a review in one of the journals. We summarized the periodontal cell-derived EVase in periodontal treatment. Very interestingly, we found that jawbone or velobone-derived osteoblast, I'm talking about primary cells, jawbone-derived osteoblast-derived EVA are very less studied. I'm not sure the reason why. However, the role of mesenchymal stem cells, bone marrow-derived or adipose tissue-derived or dental pulp derived stem cells, derived EVA, have shown can significantly promote periodontal treatment. Those stem cells from other tissues or even from dental pulp, they have their own, specific signatures. They cannot capture the local periodontal tooth root structure. So from our perspective, we assume that our periodontal T cells derived EVA may be more optimal for periodontal treatment. That's one of my PhD students' project at the moment, that comparing 3 different cell type derived EVA in periodontal treatment. Those 3 types are from periodontal ligament cells, jawbone, alveolar bone derived osteoblast, and gingiva derived gingiva fibroblast. This project is still in progress. Hopefully, we will see the results in next year also. Here you are. From my own knowledge, I feel extracellular vesicles in salivary or GCF, or extracellular vesicle from cells, different cell type, they are powerful diagnosis and therapeutic in periodontitis diagnosis and treatment. That's my understanding. This is a great question. Research goal, as I mentioned, we mainly focus on EVase to diagnose and treat the periodontitis. Of course, in the next 5 years, I hope my research for diagnosis, I can develop a very easy use for patients or for the clinicians to use off-shelf or chairside diagnosis tool. For example, similar thing like our rapid antigen test. That's my dream or research goal, that my diagnosis study can reach that stage. But be realistic, I don't think it can reach there in 5 years. Maybe in 10 years, potentially it can reach there. At the moment, we are still doing fishing to see, hunting around to see which one is the best marker. And then we found a biomarker panel. Ideally, the biomarker panel can be used in same panel or same microfluidic. For example, we can use microfluidic technique or lateral flow assay, LFA, or combination using microfluidic or LFA device. In the end, we can see, okay, this person has stage 1 periodontitis. Okay, time for you to see your periodontist. Or when some periodontitis patient, during their treatment, we can follow up, okay, during this treatment stage, your periodontal health can reach healthy or less severe state. This is a little bit ambitious. It's a long way to go. However, this is one of our research goal and a research interest. It's my own and my supervisor's research goal for diagnosis. For the treatment side of things, EVase, as we know that EVase has been utilized in human clinical trials to treat a lot of disease in the field, cancer, this and that. I hope at the same time our therapeutic role of EVase can be progressed in small animal model, then large animal model, or even in phase 1 clinical trial. Hopefully. Again, I don't think it will happen in 5 years, but that's our research goal. Because EVase therapeutic, everyone knows that it has a lot of challenges and there's no standardized protocol how pure our EVase should be for treatment. I hope our EVase field can progress further and we follow same protocol, make it standardized, make it clinical grade, clean grade of EVase. Yeah, to summarize, my research goal is in the next 5 years is we expect to potentially reach a commercial product to diagnose periodontitis or closer for clinical trials for treatment. Basically, this question also equals to what do you want to say to your young self? Yeah. Everyone has their own experience, career path. From my own path, if I have a chance, I would talk to myself during first start of my PhD. The first one advice I would tell myself or other new young scientists is grab every opportunity that you can, especially during your PhD. For example, in a PhD, stage, most of our PhD students are focusing on lab work, go to lab 24/7. No, not 24/7, that's too much. You work hard in the lab, and then you ignore everything else. For example, grant application, and some other service community roles. You just out of the scope, and you have no idea in the future, okay, service to the community is a critical part in your grant application, especially in your PhD degree stage. You can have a chance to apply for a grant. Doesn't matter how big the grant is, just grab it. It's a great opportunity for you to practice your grant writing skills. Grant writing and paper writing and report writing are totally different. So first advice I would tell young scientists is grab every chance during your PhD study. Second advice will be for those people who graduate from their PhD. For example, you just finished a PhD and you're confused what you're going to do. That's a critical turning point for every PhD students. Or after study, you need to decide you want to stay in the academic or you want to go to industry. I only can give advice to people who want to go to academic because I'm still in the academic. For those people who want to go to academic, I strongly suggest you talk to your senior postdocs or senior colleagues, or especially to your supervisors who are very experienced on fellowship grants. In Australia, grant opportunities are relatively better for ECR, which is early career researchers, for those who graduate less than 5 years. Once you miss this, you have very less chance for grant or fellowship. This is a second advice to the people that when you graduate from a PhD, advise your supervisors and senior colleagues to ask a potential pathway for you to get a smoother academic. Because in my own experience, I missed one of the great grant opportunity. Year one, I think after I just finished my PhD, I had no idea what's going on. And at that time, you only can apply for one ECR grant, only twice, only 2 chances for life in the first 2 years. I missed the precious first time. So I didn't go for the grant application in the second round because I have no previous experience in writing. So in my second round, my grant didn't go well. So practice can make it better, of course. So second advice for the ECR would be, again, is seek for help, grants. Yes, I'm talking about grant into first and second advice because grant matters. Without grant, you can't do any research or you even can't sustain your own job. That's number 2 advice. Number 3 advice, I would say that, again, from my own experience, you need to find a good supervisor to support you. It's not always the case in the academic field. Most of the supervisors are very busy. They have their own goals, and your goal and their goal may not match. If your supervisor is supportive for you to go for fellowship, grants, paper writing, then you are very lucky. However, sometimes your supervisor doesn't support you enough. It's not a good sign. It's a good sign for you to leave and find a proper supervisor. So number 3 advice is to find a good fitting environment for you to grow your own academic career. I think that's most of it. If I can give more, that will be as ECR or mid-career researcher. If I can give 4, number 4 will be you better practice your supervision skills as early as possible, because supervision is not as easy as mentor. Mentor is hard to describe. A supervision, a PhD student or master student or honor student or undergraduates, they are different levels. Then you need to set up different expectations to be clear what you want. And yeah, practice your supervision skills under your senior supervisors and senior colleagues. Start as early as possible, because everyone needs to learn how to be a good supervisor as well. That's all, I guess. To summarize, the first advice will be grab every chance you can during your PhD candidature. Second will be seek for help regarding the grant opportunities just after your PhD. And number three, to find a good matching environment with your good matching supervisor. Number four will be practice or improve your supervision skills slowly and start as early as possible.